Hi everyone, this is Dr. Roy from the biology department. I'm the resident plant biologist of the department, so I thought I'll make a video to show you some of the things we can do with plant growth experiments that we have been planning as part of our FB1 lab. So one of the things I wanted to show you today was some of the data that you can collect from these Wisconsin fast plants that you've been growing. So when you have plants that have been planted in these quads, you see how they start germinating and start forming little seedlings. So one of the first things that you can do is that we can count how many seeds germinated. So germination is the process by which a seed becomes a shoot and a root. So you can see that every seed will produce a structure such as this, which is called a seedling. And if you have planted 8 seeds and you get 8 seedlings, then you know that your germination is 100% complete, where all your seeds germinated. Now, if you planted 8 seeds and you got only 4, then you know that your germination rate was 4%. So boom, you have your first data that you can collect from the first week after you've started your experiments. Some of the other stuff that you can do is you can have a ruler, you can take a ruler and measure the length of these seedlings, and you can also measure the width of the leaf length. So you can basically measure this distance right here between the tips of the leaves here. So this gives you the leaf span, or how much have the leaves opened up. And this can be a really important data for you to collect if you are studying the effect of temperature or the effect of light on leaf growth. We also have these digital calipers, which are much more accurate. So if you are struggling with the ruler, you can always use these contraptions here, which give us a digital output of what's the width or the length. So you switch it on and you zero it. And then using this knob right here, you can open up the calipers. And for example, if you wanted to do the leaf span, you could stretch it out such that it touches the edges of the leaf. And once you've done that, you get an output that shows you, okay, the leaf span was 10.74 millimeters. So similarly, you can do this for multiple seedlings and collect the data as to, you can collect the data on multiple seedlings and write them down. And so this you can do over multiple weeks to see how are the leaves opening up. You can also use the digital calipers the first week to measure the length of the seedlings. But eventually, since they will grow taller and they will start looking like this, you're probably better off measuring with a ruler. Now, there are a couple of things you can do when the plants start to look like this. You can measure the number of leaves on the plant, the number of flowers on the plant, and be very careful to collect the data every week, because if you miss a week, you will not be able to find a trend in the data. So the whole goal is to find out trends in the data as to how your experimental group and your control group differ. So if you have 2 leaves the first week and you then find 3 leaves, 4 leaves, if you miss a week in between, then you're missing that trend of how the leaves developed. Measuring the number of flowers is also very important. It can give you an idea about the ability of the of the plants to reproduce and produce flowers. Some of you might be doing experiments where you're checking the effect of temperature or the effect of light on plants. And one of the key features that becomes variables in some plants when their conditions change is how soon does the plant flower. So the time taken for the first flowers to appear on the stem can be a critical feature that you can measure. So you can basically measure after 2 weeks, if you see that one of your treatments has flowered, whereas the other one hasn't, you can make a point. Or if you see 3 flowers open, for example, here you can see this is an open flower. Once you see the buds have opened up and produced the yellow petals, that usually suggests that the flowers have opened. So you can count the number of open flowers on a stem. The other thing that you can do, which is really cool, is Once the flowers have been fertilized, you can get these little structures known as siliques, also known as fruits. And so I'm going to pluck this one off and show it to you up close. So these are siliques. And inside them, you get seeds because the ovules have been fertilized by the pollen. And so you could measure the length of these fruits while they are still attached to the plant. And you can measure the length of the silique. You can measure the diameter of the silique. This gives you a good idea of how the silique is developing in its width and its length. And also, you can, after your experiments are over, you can open up the fruits to see how many seeds are in there. And this can be done with a razor. I am lazy, so I did not get up and try to find a razor, so I'm going to use my fingers to open this up. And once you do open it up, you will see that inside this, there are these little seeds that are developing. And anyways, I'm not gonna try to do this on the video. If you do need help measuring seeds, let one of us know, and we will show you how to slice it open and measure the number of seeds that are in there. There is another thing that you can do with your experiments at the end is you can take, so once you have measured the height of the plant, once you've measured the, there's one more thing you can do is you can measure the diameter of the stems. So each of these stems has a certain thickness to it. And so you can grab it with the calipers and measure the thickness, and it gives you a readout. So this kind of output also tells you whether a plant is really happy and growing nice and wide, or is it becoming spindly and narrow because of stress conditions. What else? So you can do leaf. You can also do leaf shading. So if you have leaves that are stressed, you can place them on this shade chart here. And you can make a point that, this was in more between 5GY 712 and 5GY 610. These are paint shade cards. Some other techniques would employ using digital software that measures the amount of pigment, but we are not going to do that. And compared to that, if you have a leaf that looks a different shade of green, so for example, if you have these two leaves and you want to compare them, you can always put them on the shade card and say that, my experimental group had much more yellow leaves that were stressed. So we've covered germination, we've covered the number of leaves, the number of flowers, the time to flowering, the length and the diameter of the stem. We've also measured the length of the fruits. We also talked about measuring the number of seeds inside the fruit. And finally, if some of you are interested in some destructive sampling, once your experiments are over, you can pull out the plant and you can see that it has a beautiful root system that is forming here. So you can see the roots are coming out. So you can shake off all the soil, take off the stick and shake off all the soil. And this is going to be a little bit tricky, so be careful not to rip up the soil because the root is in there. But basically you will be able to start seeing the root system. And you can make measurements on the depth of the root system. So if the root system has completely come to the bottom, you can measure it and you can find the length of the root system. And the other thing you can do is you can also see if I will help you out if you need help with this, but basically I'm trying to find lateral roots, but let's not worry about that right now. But the length of the root could also be a powerful thing to study. So you can see this whole thing is the root, and you can see the end of it is coming out here, so you can measure the length of the root. Okay, so that is another aspect that you can measure if you're interested. But besides that, if you come up with something new that you want to measure, you're more than welcome to. We are very excited for you to do these experiments. And we are very excited for you to collect data on something that seems very simple, but can be a very powerful tool for understanding different kinds of effects on the biology of plants. So hang in there, collect some data, have fun. FB1 is proud of you all. Thank you.